global perspective? I'm learning before that we got here, sometimes you can use triplet therapy, but for the most part not yet approved. Sanjay, maybe we can start with you. What's going on in the UK?
It's quite simple. So the 189 regime has only recently been given a European label. So there will be country-specific reimbursement discussions. It's not routinely used in the UK because it's not reimbursed. And so we're very much looking forward to having this available and making this a treatment option for our patients.
So your sequencing strategy for a patient in the UK who's advanced adenocarcinoma without an actionable mutation would be carboplatin, pemetrexed followed by immunotherapy?
No, so we have access to Pembro monotherapy in
if
the 50% plus group.
Less than 50% though?
So they're getting platinum doublet as standard with a view to immunotherapy on progression at the moment.
Okay. Same in France?
This is where we are in France currently, so that means it's not already refound for the combination for the patient lower than 50%, but for sure that it will be the standard of care in this population with a low PD-L1 expression. So we'll keep the Pembro alone for the more than 50%, and lower than 50% it will be chemotherapy and pembrolizumab. Okay. So it should become shortly, but until now it's not already refound.
And in Switzerland?
So in Switzerland, it's still not registered because we have our own regulatory authority, right? But it's going quite fast. But we have access in our compassionate program. So it's now since the beginning of the years that we can use it, at least for the 189 data. Okay. So we're using it routinely. But I'd like to add just some nuance about negative, the lowest, or even the more than 50% PD-L1. I had this patient, I had a second opinion by email from a colleague telling me that he has this BRAF never smoker positive patients with this PD-L1 of 90% or almost 95%. And was asking me, okay, the patient fears to get toxicity, but he's a never smoker with a BRAF mutation. Should I give targeted therapy, monotherapy Pembro plus chemo? So still, there's a gray zone where, I mean, everyone is right. Yeah. We don't know for these never smokers, it might be with low tumor mutational burden, maybe where if you have a threatening disease, lots of disease, and a never smoker status, maybe some chemo might be a good idea. So maybe we individualize a bit more the treatment strategy there. And on the other hand, if completely negative PD-L1, completely negative, I don't like the PFS curves. The PFS curves are not good. So maybe here you should consider some other opportunities for checkpoint, checkpoint, So there will be this debate. It's really kind of instinctive and opinions, right? Yeah. But I think there will be these marginal cases still opening the debate at the tumor board.
Right. What I would say is, Solange, I agree with you. These decisions are very difficult, and when the patient is sitting in front of you, the considerations are often unique and draws a lot on your experience. For patients with driver mutations, though, which we will talk about in a second part of this conversation, I feel that targeted therapies provide the best outcomes possible. We know that for EGFR to a good extent, and I would venture to say that even for BRAF and ALK positive patients, what we see with targeted agents now, with more and more exciting agents coming down, they should get the targeted therapy, and when we run out of targeted therapy options, we move in immune checkpoint inhibition, no matter how high the PD-L1 expression is.
Yeah, I think there's a lot of unanswered questions about the role of immunotherapy in specific genotypes. I think we have good data with single checkpoint blockade in the EGFR and ALK space. We're learning more and more about these other actionable yet rare mutations that we're finding. And I think there's a lot of unanswered questions about how to move forward. I agree, Suresh. I think that targeted therapy is the best chance for these patients, and it really speaks to the whole idea that we wrestle with in the US of tissue stewardship. We send off the tissue, we get a PD-L1 right back within a day, but the NGS is taking 2 weeks, and you may be fooled on exactly what to do. So I think it's a real challenge. David?
That's why, for me, PD-L1 might be a good biomarker, but not a good biomarker alone. So that means if you don't have the other molecular testing for the EGFR, for the BRAF, for the ALK, and also for the ROS1, I would never start an immune treatment alone, and particularly in a never smoker. So that's why for me, PD-L1 is a good biomarker. The smoking status is probably the best biomarker. If you have a never smoker in front of you, you might fight to find a molecular alteration, because you can't find a molecular alteration, and this is a priority. And in all the trial, in fact, in never smoker, you do not prove completely that an immune treatment do better than the other treatment and chemotherapy and so on. So that's why for me, in these cases, before to start any immune treatment, try to find a molecular alteration. And if it's BRAF, as you told Solange, clearly a BRAF mutated never smoker probably will not benefit of an immune treatment alone, will have a major response in terms of PFS response rate with targeted therapy. So that's why for me the smoking status is something quite important, particularly in first line.
It's even more fragile in EGFR, this story of having really the whole picture, and you need to know, because we know from a very recent publication in JTO that monotherapy checkpoint doesn't work and even is risky for this patient. This patient die before seeing the other treatment. So if you had to ignore an EGFR mutation or not know about it and decide for the triplet, you go back to the old days where you give chemo versus TKI and you give the chemo, which is not the good decision. And the other way around, Matt Hellman was telling yesterday from Sloan Kettering that he had also some time to catch up with a late discovery of an EGFR mutation. But now we give osimertinib frontline. So you have a checkpoint on board of your patient with a very long pharmacokinetic, and then you have to give osimertinib on the top of it, and nobody wants to do that because it creates pneumonitis. So have the full picture, and then you have to make a decision also with some discussion with your patients, I guess.
I agree, because even if you start the immune treatment and if you stop because the patient progresses after 2 or 3 cycles, you will start a targeted therapy. In fact, you will still have a huge amount of antibody in the blood of the patient, and they might develop some specific toxicity. So that's why it's another issue. So even if you think you have stopped the immune treatment, you start an EGFR TKI, a BRAF inhibitor, and so on, if there is some interaction with an immune treatment, you will develop the toxicity, because it's at least 4 weeks, 6 week half-life with an immune treatment. And you cannot stop for 2-3 months before to start any specific targeted therapy. So that's why we have to take in account, and it's completely something new. And we should keep in mind also this type of toxicity after the immune treatment, even if you stop early the immune treatment.
Very, very good point.